Okay, so here is a review of AP Bio and Unit 1. It includes all of the essential knowledges listed in the course curriculum. Okay, so the first one says the subcomponents of biological molecules and their sequence determine the properties of that molecule. So when we look at biological molecules or macromolecules, or organic molecules, they are talking about carbohydrates, lipids, proteins, and nucleic acids. So while water is also included in Unit 1, macromolecules or organic molecules are a big part of the standards in this section. So when we talk about the subcomponents, those are going to be your building blocks, your monomers that will join together to form your polymers. Now, when we look at, we can go back. When we look at the sequence, when it says their sequence determines the properties, well, that sequence could be in, like, for example, if you look at proteins, the sequence of the amino acids will determine its properties. And in nucleic acids, the sequence is the A, T, C, G, C, A, C, etc. Okay, so let's go ahead and talk about water for a little bit. So it says living systems depend on properties of water that result from its polarity and hydrogen bonding. So two key things about water is we want to make sure we understand why water is polar and how that polarity leads to hydrogen bonding. So when we look at a water molecule, it is made out of an oxygen with two hydrogens. Now that oxygen right here is this oxygen is super electronegative, which means that when it's sharing electrons with the hydrogen, it has an unequal, it's like stronger at pulling the electrons towards its nucleus, which then basically leaves the hydrogen side of the water molecule with a partial positive charge. This is not the same positive like a sodium ion, but it just means that that region of that water molecule is slightly positive, where the oxygen side of the water molecule is going to be slightly negative simply because you had that uneven sharing of those electrons. So now when we look at those slightly positive and slightly negative sections of a water molecule, we can see how they interact with other water molecules. So here in blue, you have the slightly positive hydrogen, and then in green, you have your slightly negative oxygen. It is that attraction between these two molecules, the slightly positive hydrogen and the slightly negative oxygen, that we call a hydrogen bond. So this one is pointing to the hydrogen bonds we have here. Okay? All right, so the hydrogen bonds between water molecules result in cohesion, adhesion, and surface tension. So you can see again here you have a slightly positive hydrogen ion and a slightly negative, a slightly positive hydrogen atom that's part of a water molecule, and a slightly negative oxygen that's part of a water molecule being attracted to each other. So when you have two water molecules attracted to each other, that's going to be cohesion. But when you have a water molecule slightly attracted to something that is not water, we call that adhesion. So you experience adhesion when you get out of the shower or out of a pool and the water is sticking to you. That is adhesion. But water, attracted to other water, is going to be cohesion. When we talk about plants, cellulose is slightly polar in some areas, and so therefore the water being attracted to cellulose would be adhesion. Now here, water attracted to other water molecules is going to be cohesion, and that creates a surface tension that's hard to break those interactions between water molecules, and allows lightweight organisms to kind of walk on water, for leaves to float on the surface of water. So when I look here, I want you to think about which section is showing a hydrogen bond. That'd be C. Which one is adhesion? That's going to be D. Which area has a partially negative area, that'll be E, and a partially positive is going to be A. Now this one, though, we haven't really talked about yet. Right where you have, if we go back to this, let's go here. When we look at the bond between hydrogen and oxygen, that's a covalent bond between the oxygen and the hydrogen, but it's a polar covalent bond. So it's covalent because they're sharing electrons, but it's polar because oxygen is like pulling those electrons. Okay. So let's go ahead and talk about this next standard that talks about organisms must exchange matter with the environment to grow, reproduce, and maintain organization. So yes, we have the water cycle, the carbon cycle, the nitrogen and the phosphorus cycle, but really what we're talking about are the 5 major elements that make up life on Earth. We have carbon, hydrogen, oxygen, nitrogen, and phosphorus. And these elements are going to cycle between living and non-living things on Earth. For example, as I'm talking right now, carbon dioxide is coming out of my mouth into the air, and then a plant will take it in by photosynthesis. And maybe it'll be a carrot plant, and then I'll eat that carrot. Now it's back part of me. My mitochondria will break it down. So these elements are going to go through cycles, and they're not created or destroyed, but rather just transformed on Earth, and it's a beautiful process. So when we look at the atoms that are necessary, atoms and molecules necessary to build macromolecules from our environment are the carbon, hydrogen, oxygen, nitrogen, and phosphorus. Now, it is important to remember that carbon, hydrogen, oxygen are found in all 4 organic molecules. However, nitrogen is found in proteins and nucleic acids, and phosphorus is found in just nucleic acids. So that is something to kind of try and remember a bit. And so when we look at this, nitrogen is used to build proteins and nucleic acids, and phosphorus will be found in nucleic acids. Now, the one place you will find phosphorus in lipids is going to be in a phospholipid as part of the cell membrane. So when we look at how we can join the building blocks of life, like the monomers into polymers, we have what's called hydrolysis and dehydration synthesis. So hydrolysis is going to be where we're going to add a water. So right here you can see in this GIF, you add a water, hydrolysis, and it causes it to split. Whereas dehydration synthesis is removing a water to form a bond. And so when we look at hydrolysis and dehydration synthesis, you do see it in the ATP to ADP cycle. So when we talk about using ATP and the energy stored in the bond, it's actually hydrolysis that's going to add a water to break off that phosphate group. And then to attach a phosphate group to an ADP, you would actually form a water in dehydration synthesis. When we eat carbohydrates, you can see in this GIF here, when we eat carbohydrates, let's say you have some sugar. This is a picture of sucrose, which is your white table sugar. When you eat that, you actually will have an enzyme, salivary amylase, in your saliva that's going to add a water to separate those 2 monomers, 2 monosaccharides. So here we formed a water. And then when you add the water back during hydrolysis, that's going to break the bonds. Now in carbohydrates, this bond that's forming right here is called a glycosidic linkage. Okay, so when we look at Unit 1, it also includes nucleic acids. I like to focus on nucleic acids in Unit 6, but here they are also made of CHONP in a monomer-polymer fashion. They will use dehydration synthesis. So when you attach macromolecules or nucleotides together, attach nucleotides together in that sugar phosphate backbone, it will use a dehydration synthesis reaction in order to form those bonds. Okay, now let's go ahead and talk about proteins. So when we talk about proteins, their monomer is an amino acid. And in an amino acid, you have an end of the amino acid, which is your amino group, your amino like terminus. The word right here, terminus, means like end. And then you have your carboxyl end. Now, when we look at an amino acid, all 20 amino acids have the same structure. The only thing that's different will be their R groups. These R groups will vary from one amino acid to another. Some of them will have like polar, nonpolar, positive, negative properties, and that will determine how that protein folds up. So when we look at a protein and the steps in folding, your first step in folding up of a protein is the sequence or the order of the amino acids. Let's see if I have I don't. Okay, so when we talk about the polypeptide here, the sequence of amino acids is the primary structure. Then in the secondary structure, you have the alpha helix and the beta pleated sheets. Those are going to be due to hydrogen bonds between the backbone of the polypeptide. Then in the tertiary structure, here you have the tertiary structure. You start to have different kinds of interactions together. In the tertiary structure, you will have nonpolar amino R groups interact with other nonpolar R groups. You'll have like ionic bonds form between R groups, etc. So you start to have a folding up of that protein, and then in your quaternary structure, that'll be multiple polypeptides joining together. Now, when we look at the structure and function of polymers, they're derived by how their monomers are assembled. So when we look at carbohydrates, carbohydrates have different structures depending on how they're formed. So when you look at cellulose, cellulose is going to be very straight chains, very difficult to digest. Really just bacteria are able to break these bonds in cellulose, where starch is made of all alpha glucose molecules. And cellulose is made of alpha, beta, alpha, beta, alpha, beta. And then here we have some glycogen. You can see like a branching shape that we will break off in our own bodies when we have low blood sugar. So when we look at lipids, so lipids have different functions in our bodies. So lipids, if we talk about a phospholipid first, a phospholipid has two different regions. You have your like your phosphate group, let's go like here, and then you have your fatty acids. So in a phospholipid, you have one area that is hydrophilic and another area that is hydrophobic, and this forms the basis of our cell membranes, but we'll expand on that in Unit 2. And then, so here you have your polar head and the nonpolar tails. Now, our nucleic acids, I'm going to talk a bit more in my Unit 6, but basically they have a 3' end and a 5' end. You can see right here they're going to have a phosphodiester bond form by dehydration synthesis, and then they'll have hydrogen bonds between their base pairs. All right, so directionality is referencing the 5' and 3' ends. Nucleic acids run anti-parallel to each other. And then we have our proteins. And in our proteins, the bond that connects two amino acids together is a peptide bond. So a peptide bond is going to form between, let's pick up this one. So here you'll have the amino group of 1 and the carboxyl group of, did I write that right? I don't think that's right, but that's okay. The carboxyl group of another. I don't think that R group is supposed to be from there. This figure might be off, but that's okay. So let's use this one down here. So here you have your central carbon. Here's your carboxyl end and your amino group. So you can see in amino acids, you have the carboxyl and the amine group are what join together by dehydration synthesis to form that peptide bond. And so when we look at the folding of a protein, you have your primary, secondary, tertiary, and quaternary structures. And then here, DNA and RNA, we'll talk about in Unit 6.